Hi, I'm Alexander and I'm one of the co-founders here at SynBioScene. Today I'm really excited to show you a conversation that I've had with someone very exciting from a SynBio perspective. And that person is Sarah Holland, who is not in the SynBio sector anymore per se, but she used to be at Lonza, the global CDMO here in Switzerland, where she was responsible for the global scouting and evaluation of new technologies that Lonza could use in their manufacturing. And she was also running their bio R&D for some time, and she was on the board of Bacthera, a Swiss SynBio company. And I'm really excited that I can show you this today. I hope you enjoy.
So let's dive into the topic really of today, and to start out to really kind of define what we're actually talking about. There's many different ideas on what exactly synthetic biology is, but in your mind, how would you define that topic? How would you say this is synthetic biology?
Well, it's interesting because when I was at Lonza, I sort of built this R&D leadership team, and then together with a really good consulting company called PA Consulting, we specialize in technology, we actually looked at synthetic biology in quite a structured way, and we started off by defining it. So we said it was an engineering approach to biology with the goal of creating tools that allow design and build of functions in cells. So that was how we defined it, which kind of was similar to the SynBioBeta and SynBioScene definitions. It's all about engineering biology.
Okay. So then just to kind of discuss this a bit, I mean, so you would say if you take the human insulin gene, you put it into bacteria, is that already synthetic biology? Because is it really design already?
I think we've been doing synthetic biology for some time, but we've only really kind of woken up to it. And in fact, I always think it's quite funny because we think we invented, standardized manufacturing, but actually nature invented it. That's what DNA does, So synthetic biology has been around for a while in biologics manufacturing, for example. But I think now the tools have started to come together to really do it in a thoughtful, purposeful way rather than kind of a serendipitous discovery way. So the ability to do it has really mushroomed. And we didn't only look at the kind of biological gene editing side of it. We also thought that for us, certainly as a CDMO, a contract development and manufacturing organization, it was the miniaturization, it was the design, build, learn element, it was the artificial intelligence, the computing, it's all of it together. It's not just having gene editing tools, it's also having miniaturization, high throughput, design, build, learn, computer-aided design, all of that stuff coming together. And so we started off with a definition, and then we built what we called a taxonomy, where we broke it down into different areas and put different levels on them. So we ended up with a kind of picture of what we thought synthetic biology was that was quite broad.
So you're saying, kind of, I'm paraphrasing, but a scientist in a lab, engineering an organism is, there has to be kind of more parts to that. That's what then, becomes synthetic biology, when everything kind of comes together, really.
Well, I think anybody in a lab doing, engineering a cell is doing synthetic biology, but to do it at scale with impact, it really helps to have all of these tools we identified some companies that we described as integrated, who were able to take, because there are many companies who specialize in one thing, like computer-aided design or, web-based experimentation, where you kind of switch things on your laptop and a lab somewhere on the other side of the world does the experiment. There are all these different technologies available to you, but some companies actually integrate everything into a platform, and that's when you really start to see the magic,
Okay, got it. I think I have an interesting anecdote that I wanted to share, because when I was a student at the University of Zurich, I was doing an internship in a lab for plant biology. And at the Christmas dinner, I sat next to the professor and I asked him, what is actually going on with synthetic biology in plant science? And he looked at me and he said, well, we've already been doing it, and there's another word for it. It's called, bioengineering. And kind of, why is there a new term now for, I mean, that's what he would say. Why not just call it bioengineering?
I don't know. I mean, I think plant science, that there's been conscious development of traits in crops and an understanding of jumping genes and things like that. So plant science almost led the way in lots of ways in this whole field. I don't know. I mean, I think what you call it isn't probably the most important point. But synthetic biology seems to be sexier, and you can shorten it to SynBio, so maybe that's why.
Yep, that's a very good take. I like that. Thanks. So, did you still remember the first time you've heard about the topic?
Formerly, synthetic biology was probably a colleague of mine who introduced me to it at Lonza. He was very kind of, some people, I think they call them, in management research, they call them lightning rods or something. There are some people who are really good at spotting things going on in the outside world and bringing them into companies. And I think he was definitely a lightning rod. He's called Ryan Scanlon. And I brought him into my business unit and gave him quite an entrepreneurial, creative role because I could see that was his phenotype. I think he was the one who had a deck about synthetic biology that kind of just got me excited about the whole topic.
Okay. And what was your first impression when what were you thinking when he came to you with this term, this new idea?
I just thought this could be amazingly important for Lonza. We needed innovation. We needed to match up customer challenges and what we needed to offer customers in terms of value creation with what this technology could do.
Right.
So in this project I was talking about, not only did we look at kind of what's the taxonomy and what are the different building blocks, but we also looked at what does Lonza need to do better. It was things like complex protein manufacturing. Proteins have got, biologic drugs have got much more complex. They're bispecific, trispecific. They're very squirrely things now and very hard to make. Viral vectors are incredibly hard to make. That's a kind of third order problem. Continuous manufacturing was another big challenge. And then, potentially Lonza could help with therapeutic design. Like, how do we help our customers avoid immunogenicity and, other things that trip them up? It's all about getting drugs to patients faster. But you'd be amazed how important the manufacturing step is in biologics and how that slows you down and messes you up. And if we can offer, obviously I'm not with Lonza anymore, but if we could improve that and offer it faster, more effectively, with better results, better yields, that would make us, competitive in the industry. So it was seeing that opportunity to really impact customer offerings that was so important.
And do you think Lonza is early when it comes to adopting more of these approaches, or is it, if we look at CEOs in general, do you have an idea of how common is it already to really say, okay, synthetic biology, this is kind of the next thing for manufacturing things like biologics or gene therapy?
I think Lonza was a little bit challenging to bring innovation into, because if you think about it, most of your colleagues are working in the operation side of things, doing GMP, and the last thing you want to do in good manufacturing practice is change anything. You want to do everything perfectly the same each time. Don't mess with it. It works. So you're trying to say to people, I want to disrupt what you're doing and have you do it a different way that's going to cause, you're going to have to invest time and learning and skills training and equipment and all this stuff to do something better. So what I've always found, when you want to bring innovation into an organization, you need to kind of get in there under their skin of the people you're trying to influence.
Right.
And so what keeps them up at night? What's their biggest problem? And it might be something small to start with, but you try and solve their problems and show them that you can add value. And then you open them up to being open to new ideas and doing things differently. And then you can gradually get more and more disruptive once you've shown that you understand their problems. So it was all about, and complex protein manufacturing was a good hook, because, if your customers are having problems, because often with a cell line development, it's only quite late on that you discover your cell line is not stable. And that's really serious. You've got to go back to the drawing board. So if we could improve some of the problems that were really causing customer issues and problems, then people would start listening to us. So one of the things I did early on was to bring in the piggyback technology, which was the jumping gene technology, to improve cell line stability and the ability to manufacture complex proteins. And when that started to actually deliver what I promised, people start to listen to you.
Can imagine. So results first, that usually works well with the scientists. Good. Okay. So perhaps for those people who aren't, maybe university students or at iGEM, what does it actually mean to be a global head of licensing at a company like Lonza?
Well, it never existed before, so I was able to kind of define that. So the interesting story, I guess, is that Lonza acquired Celltech in the UK in the early 90s, maybe late 80s, quite a long time ago. And they were pioneers in developing the GS cell line, the CHO cell line that's used for lots and lots of biologics. So the licensing that technology because we didn't only manufacture in-house, we also licensed the technology to enable it to be used outside Lonza in other companies, even by sometimes even by other CDMOs. But that was quite an old technology. So we had this wonderful kind of, cash cow of, money coming in from royalties from enormous drugs that were on the market. So that was great. And I could have just gone to the beach with a beer and collected the money. But the worry was, this is very old technology. We need to improve it. We need to keep it relevant. We need to innovate, So that was why I got so enthusiastic about innovation, because we needed to find ways to make GS, modern and introduce things like piggyback, look at other technologies. We had a really interesting relationship with Harvard on a viral vector platform, which is actually about the design of viral vectors. So, what can we do with that technology, for example? So, yeah, I could have just collected the paychecks, but if the feeling was this isn't going to have a long-term life as a business unless we innovate.
Right, and the innovation was clearly with I'm paraphrasing from your LinkedIn profile. I think you said, emphasis on new technologies like gene therapy, synthetic biology. That was kind of what you're saying is the future. Yeah.
Exactly. Yeah.
Okay. And so, and you had, I guess, a team below you who would be more doing the groundwork of scouting new opportunities, and you would kind of evaluate them, or were you also, scouting actively, or how did it work?
Well, I guess the team was quite small, and we had a global, business unit. So literally, we had one person per continent doing out there doing
exciting
sales effectively, and then we had negotiators, and we had marketing and so on. But it was very, very small, so there weren't many of us. So in terms of scouting, I guess Ryan and I were probably, and obviously you tap into the R&D folks because they bring great ideas. So created a strong relationship with R&D, which probably is what led to me then taking on the head of R&D role on top of the day job kind of thing. But yeah, I guess you have to be open to wherever an idea comes from, but typically it'll come from R&D or your kind of lightning rods like Ryan.
Right. And how did now you started talking again about the R&D role. So tell us about that. How did this happen? What did this mean for you? And just give us a bit of an insight there.
It was very funny. I was invited. The head of R&D said, "Can you meet me at the Basel SPB train station at 4:00?"
Okay.
And I said, "Okay." And I turned up, and he said, "We'd like you to start running R&D from Monday because I'm leaving." And I said, "Well, if you buy me a grappa, we can talk about it." So we had several grappas. And so this was May 2019, I guess. Yeah. So it was a bit of a surprise. It wasn't planned. I guess he'd, got something else he wanted to go and do. And I guess they must have seen something in me that they felt, because I was very surprised. And obviously it was on top of the day job, but it was fantastic because it was a wonderful chance to really understand everything that was going on. And Lonza had, maybe remember this one, it seemed like about 600 people across 13 sites or something doing R&D. It was a lot of R&D going on, very scattered but very interesting. So it was a chance to build a leadership team, create areas of centers of excellence in different sites, introduce everybody to synthetic biology and other novel technology, start to open their minds to what was going on outside, really sort of prioritize who we should be partnering with, and also encouraging people to buy modern equipment like robots, and really kind of up our our level of functioning. But I was only doing it for a relatively short period. I think it's about 9 months, and I got somebody much more confident who knew what they were doing to take over from me, Katerina Radozovic, who
Right.
was heading up biologics in Sanofi. So I was very glad to find somebody that great to take over. But it was a wonderful opportunity to really, tighten focus, not upskill, but kind of bring people into the modern day in terms of what's going on and and how we should be tackling problems and what problems we should be tackling, and sorting out where we focus the budget and what our goals were and all that stuff.
Right. Okay. Yeah. Thanks for expanding on that a bit. How did this then? I mean, how did this relate to the synthetic biology? I mean, you're saying that you were closer to the pulse, let's say, of the R&D people, and you kind of used that to leverage then your ideas for what you wanted to do.
Yeah, I think Ryan had kind of made me aware that there was so much going on, and also doing the piggyback deal had kind of opened my eyes to the power of these technologies to really solve our problems. So then
Right.
I thought it made sense to do this project with PA Consulting, where we really got the leadership team all participating and took this kind of fairly structured approach to try and understand what are we talking about, what problems are we trying to solve, how do we put these two together. So then we identified these key problems. I think it was complex proteins, viral vector manufacturing, continuous manufacturing, and therapeutic design. And then we designed roadmaps and said, okay, well, if I've got that roadmap, and then that's based on understanding as a CDMO what all those steps involved are. What does it take to get to a complex protein at the end of the day? What are all those different manufacturing steps and development steps? And then where could synthetic biology help? Where could it have the biggest impact? Which companies would you then choose to work with to make that happen? So it was really a roadmap for innovation and how we could use synthetic biology to, significantly advance against our key areas of focus that we want to improve our customer offering. So, and people got really engaged, really, and I love opening people's minds up to what's going on outside and who they can partner with. And we also we not only looked at companies globally, but we also looked at academic groups as well. So it was academia, and tried to understand, where is synthetic biology happening academically and in an entrepreneurial fashion. How do we understand these companies or these groups, and how could we work with them? So I think it was great. I hope, obviously, I'm not at Lonza anymore, but I hope it led to some productive collaborations and projects.
Would certainly be very interesting also for then let's say the future synthetic biologists who are maybe now studying at university, finding a career at Lonza, because I think it's still in Switzerland, it feels like the field is still more niche. At least that's my feeling. Do you feel the same?
Yeah, I must say, I was trying to remember whether Switzerland featured heavily. I don't think it did. I mean, I think the UK came out relatively strong, because normally I think the UK and Switzerland are somewhat similar. They both have strong universities. Obviously, Switzerland's smaller, but punches above its weight. They both have strong universities. They both have quite a good environment for building startups. They're both kind of clusters, because you've got Roche and Novartis, I guess. There's a kind of a biotech cluster in Basel and Zurich. There's also around Geneva, around Lausanne. UK obviously has its golden triangle with Oxford, Cambridge, and London. But we found a lot more, well, not a lot, we found more synthetic biology companies in the UK than we did in Switzerland. Obviously, in Switzerland, you've got Selexis, who were very early pioneers. So they were an example of, a company that, they were a big competitor of ours, but they were a company that had also engineered the CHO cell, improved the CHO cell, and had an offering. So there are kind of, but I think nowhere in the world has a high concentration of SynBio companies, because it's still quite novel. probably the highest concentrations were in the west coast of the US, maybe
Yeah.
east coast, the
Mm.
Boston, San Francisco clusters. But I think Switzerland should be able to be very successful because it has all the right ingredients, the excellent universities, a really nice environment for doing startups. And I think probably more is coming. Maybe I just don't see it yet, but I'd be surprised if it wasn't bubbling up.
Mm. So that's a very interesting point, because I think I would also think the conditions are good.
Mm.
And we've done at SynBioScene, we've done a mapping of companies that we think are involved in synthetic biology, at least from our definition. And I mean, you mentioned Selectis. This is one of the companies we've also looked at. You've also been on the board of Bacthera for a while, which is
Yes, yes, yes.
Lonza,
That's very interesting. Yeah.
owned by Lonza. Would you classify them as a SynBio related company?
That's an interesting question. I hadn't ever thought of them like that. But what's the interesting story there is that obviously the microbiome's become this whole new area of medicine that none of us really appreciated until recently. Um, but if you want to sort of engineer, I guess it is engineering, you want to engineer the microbiome or influence the microbiome, you have to be able to get the bugs to the right part of the gut, which is not easy. And these are bugs that don't like oxygen, for example.
Right.
So you need the right delivery mechanism. So we had a combination of Christian Hansen, who were not well known, but actually produce all of the microorganisms used in most of the cheese and yogurt that all of us eat comes from this one company. Um, so they're really, really expert on how you handle these things. And then Lonza had this capsule that happened to release just in the right part of the gut. So you put the two together, you should be able to start really meeting the needs of microbiome. And obviously Lonza knows how to do manufacturing for medical uses, whereas
Exactly.
Christian Hansen was food. So you need to put that kind of medical lens on things. It's a good point actually, because they are extremely expert on dealing with those organisms, and I guess it is engineering. It is symbio what they're doing. I hadn't thought of them like that, but I think it is a good description actually.
I'm
And
happy
it was great
because
fun. It was
yes.
great fun to be in on the startup phase, even though it was a joint venture between two relatively large companies, but it was still had that startup feeling. So it was great fun to be involved right from the beginning.
I can imagine. I think it's very interesting to be, really there when kind of things start up and see it grow, and then that, at least to me, it feels, with SynBioScene, for example, it feels very good, a kind of very warm feeling when you look at kind of what has been built out. So
Mm-hmm.
I can relate to that. I'm happy you say, I think it's synthetic biology, because we included it in our mapping of Swiss
Good.
companies. So I was,
So we
yes,
agree.
it's good. But I mean, happy to have other people disagree on that. I think that's always a very interesting discussion. So I want to go back to, the global view though. So because you said, UK, I mean, it's interesting you said nowhere there's really a high concentration. But UK, we see more, US in the West Coast. And what about the emerging markets? What about Asia, for example?
Okay. Yeah, good question. Um, I think we didn't really, I can't remember if Asia was in scope or not. May have been out of scope, but I would say, China is just such a powerhouse now in scientific innovation generally, and particularly things like cell therapies, which definitely are SynBio, but they're more therapeutically focused. So China and India are both incredibly strong in terms of CROs, so clinical research organizations, and so I'm absolutely sure they have got things like design and build, miniaturization, high throughput, all of those things I'm sure they have. So it wouldn't surprise me at all if they took that extra step and started to build in, um, the biological part of it. Because certainly in chemistry they've been incredibly strong. But I think I wouldn't be surprised if we saw some really disruptive companies coming out of India and China, out of that CRO background. And you've also, as I mentioned, you've got the Chinese cell therapy companies who are very innovative as well. So and there's just so much money in China. It's unbelievable how much money there is. And I think they've been struggling to invest outside China recently with all the geopolitical things going on. So it's probably going back into China. So yeah, I would imagine there's some interesting stuff going on. I haven't necessarily seen it, but
Okay.
actually something we haven't talked about that was something I really wanted to do next if I'd stayed at Lonza
Yes.
was I think I've noticed GSK actually has what do they call it? They call it a synthetic biochemistry team. I think the whole of chemical manufacturing could be enormously disrupted by synthetic biology to greatly improve because at the moment chemical manufacturing is pretty dirty and nasty and polluting and bad for CO2 and all the rest of it.
Yep.
But actually, a lot of those reactions could be done at room temperature with less power, less water, using enzymes. And so I think I wanted to disrupt chemical manufacturing next. That was my target after biologics manufacturing. Didn't get a chance to do it, but I think there's huge potential there to revolutionize how things are actually manufactured chemically using enzymes, and that's synthetic biology. And there are still small startups working on that, and also larger companies like, I guess you've got the Zymogens, people like that, maybe Ginkgo are doing things like that.
I would throw Evolva in there as well. I mean, they do.
Yeah.
I mean, vanillin they do. I think a couple of other molecules. So
that's a good point. That's another Swiss. Yeah.
The Swiss
one. See, it's more than we realize.
Yeah, I think at the start we also were wondering. I mean, how much is really out there? I don't know where the mapping is at, but I think we're now finished, so to speak, with the first one at least. The sheet is quite large. I think there's more than one would think. So I guess it always depends on your definition as well. But I mean, the chemical manufacturing I think is really interesting, because also for me that's something where I also see that is from just a legal side much easier to start doing if you think about using CRISPR to edit someone's DNA. Or edit something like germ cells. I mean, that's probably still more far off regulatory wise in Switzerland than producing vanillin or chemical ingredients. I think.
No, I think you're right. Yeah, there's certainly less of those concerns. Yeah, because you're just engineering enzymes, and you could easily imagine a design, build, learn, high throughput, miniaturized, AI-driven technology platform to improve so many different chemical manufacturing processes and get rid of some of the nastier things that have to be used at the moment.
Yeah, agreed. And I think also when we talk about the emerging countries, so it's sort of middle income countries in Asia and also in Africa. I mean, when they slowly come to kind of our standard of living eventually, if we can produce a lot of commodities in a more sustainable way, I think that will be really important as well, because that's it's going to have a lot of impact as well. So just to touch on it, because you said you wanted to disrupt the chemical manufacturing next after Lonza. I mean, of course, maybe Vective Bio was just quicker, but did you already have something more concrete in mind? Where you wanted to do that, or can you share a bit more of that?
There was a little startup that I'd met. I'd gone to dinner. JP Morgan is this big healthcare conference that happened or used to happen every year in San Francisco in January. And I think I'd been invited to a dinner and sat next to somebody who wanted to introduce me to one of his portfolio companies. And I met the CEO of this little startup, and I was just blown away. He was one of those people who had 3 PhDs by the time he was 12.
Oh yes,
very intelligent. And so I went to visit them, and literally at that point it was a garage type operation. It was operating in one room of a
amazing
building in Menlo Park. And I was really blown away by what they were trying to do, and I could see the relevance to Lonza's chemical manufacturing. So then I wanted to create a collaboration with Lonza, which did ultimately happen. It was quite challenging because I didn't have the connections on the chemical side of the business the way I did in the biologic side, and there probably has been less innovation in that side of things for a while, so it was a bit more challenging. But ultimately, it did happen. And I think, I mean, this is the sort of Swiss guys at this, you're not the easiest to woo, and very hard to convince and very tough, but also very, very solid, very knowledgeable, not don't take fools lightly. And my understanding is, and I'm not in touch with it any more closely, but my understanding is it actually did deliver what it was expected to do, and they actually liked it. So that's, I mean, to get people at this, people at this
That's good to
buy
hear.
into new technology, I think is a big achievement. So yes, I did sort of initiate one little thing, but obviously the vision was much bigger than one collaboration.
Right. Yeah, I can understand that. And do you see yourself returning to the topic again professionally?
I don't know. I don't know. I mean, at the moment, I'm very happy and active and enjoying being back in the therapeutic arena. I mean, it was amazing because when I was interviewed, or when I was approached by a headhunter to do the Lonza job, he said, "Have you ever thought of working for CDMO?" And I said, "I don't even know what the letters stand for, so no, that's not something I've ever considered." But so it was a real kind of whole new sector and industry to get to grips with, but it was really good fun actually, and yeah, I think if you bring a kind of pharma partnering mindset to a CDMO, you can do a lot, So I kind of saw it as an adventure outside of my normal career in pharma, but it could be interesting to go back. There's certainly, I mean, what was great about R&D in Lonza was it went from gene editing to automation and robotics and everything in between. There's a lot more to it. And there were people working on devices as well as processes. There was just a lot more aspects to it than there is in pharma R&D. So that was actually good fun. Just the variety and the breadth was amazing.
Any last statement? Anything you wanted to say or mention?
Well, no, I think it's an incredibly exciting field, and anybody who's working in this area, I think you've got so many exciting things you can do. So just encourage you to keep, and I think one of my colleagues said that the cell is usually the problem child. So cells are they don't always do what you want. They can really let you down and be frustrating. So don't give up. It's often when you have the biggest setbacks that you learn the most and actually come up with the most creative solutions. So don't be downhearted if things are going a bit rough at the moment. I think keep going because it's incredibly worthwhile. It will make a big impact.
Yeah, that's what I call a perfect closing statement. Thank you very much, Sarah. It was a pleasure to speak to you.
Nice to speak to you too.